Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 8, Bioenergetics and Cellular Metabolism. In this lecture, we will cover the importance of energy in cells, present the concept of ATP, the energy currency, do an overview of cellular metabolism, describe glycolysis, the breakdown of glucose, present the citric acid cycle, briefly talk about anaerobic metabolism and the regulation of metabolism, and end with a summary. The importance of energy in cells. So we need energy to power movements, growth, repair. We need it to drive processes like protein synthesis and active transport. Now, the energy currency is ATP, which is short for adenosine triphosphate, and it is the main energy carrier. Now let's dive deeper into ATP, the molecule that powers cellular activities. So ATP is the energy currency. What is the structure of it? We have adenine, a nitrogenous base. We have ribose, sugar, and 3 phosphate groups. So in the name, you have adenosine triphosphate, meaning 3 phosphate groups. And as you can see in this illustration here, we have 3 phosphate attached to it. Now, how does this generate energy? By breaking the bond of the 3rd phosphate, we release the energy. So ATP becomes converted into ADP, which is adenosine diphosphate, meaning 2 phosphate groups remaining, plus an additional phosphate group. This ATP then is continuously recycled in cells from ADP and from inorganic phosphate to form new ATP. So the synthesis of ATP comes from ADP, so we have adenosine diphosphate, and an inorganic phosphate combining to form ATP. Now, the process of producing ATP requires itself energy, which is provided by cellular respiration or other energy-generating pathways, like some of these we will mention ahead, like glycolysis or oxidative phosphorylation, etc. Now, the high energy bond formed between the 2nd and 3rd phosphate groups is where the energy in ATP is stored. So when the cell needs energy, ATP is broken down or hydrolyzed into ADP and a phosphate. And this reaction is catalyzed, so this falls under catabolism, is catalyzed by the enzyme ATPase. So the energy released from breaking this high energy bond is what powers cellular processes like muscle contraction or active transport, etc. So what is important to remember here is that the energy produced from ATP is not produced from ATP itself. It is rather produced or a product of the breaking of the high energy phosphate bond between the 2nd and 3rd phosphate groups. Now let's explore how cells produce ATP through metabolic pathways. So here we have an overview of cellular metabolism. Now, when we talk about catabolism, we're talking about the breaking down of molecules to release energy. For example, the breakdown of glucose. The other one is anabolism. Here we use energy to build complex molecules. For example, protein synthesis. Now, some key pathways for ATP production is glycolysis, the citric acid cycle, or known as the Krebs cycle, oxidative phosphorylation, So to express it in a different way, to clarify the concept of catabolism and anabolism, is that metabolism itself is like a factory breaking down raw materials, that would be catabolism, and using them to build useful products, that would be anabolism. Now let's start with glycolysis, one of the key pathways for ATP production. Now let's look at glycolysis. Here you see the pathway and a lot of different individual steps described. Now, to make sense of it all, so glycolysis is like breaking a big tree log, which is glucose, right? You can see at the top here, we have glucose. And what glycolysis is essentially breaking this glucose into smaller pieces known as pyruvate. You can see it almost here at the end of this pathway, to release energy. Now, what is missing from this image is presented in the next slide. It's that pyruvate, by the use of the enzyme pyruvate dehydrogenase, can become converted into acetyl-CoA. Now, acetyl-CoA has a pathway into the Krebs cycle, or the TCA cycle. And that's where essentially ATP gets produced. But we need to get from glucose first to pyruvate. So fundamentally, glycolysis is an anaerobic process. Anaerobic essentially means that the process does not require oxygen to occur. And glycolysis is the first step in glucose metabolism, and it occurs, so this bit occurs in the cytoplasm of the cells. So its objective essentially is to break down one molecule of glucose, which is built of 6 carbons, into 2 molecules of pyruvate that have 3 carbons each. And this will lead to the production of 2 ATP molecules and 2 NADH molecules. These are electron carriers. Now, when this has happened, what happens then after glycolysis? So when we have arrived here at pyruvate, we have essentially 2 potential paths to take. Remember, to get here, we did not need oxygen. It's an anaerobic process. But when we arrive at pyruvate, as illustrated here, let me clear up the image. As illustrated here, you have 2 different pathways, one arriving at lactate and one at acetyl-CoA. So if oxygen is available, meaning we have aerobic conditions present, then pyruvate enters the mitochondria and is further metabolized via the citric acid cycle, meaning the Krebs cycle, and oxidative phosphorylation to produce more ATP. So it will then take this pathway of acetyl-CoA into the Krebs cycle. Now, this is happening. This process is occurring in the mitochondria. That's also why I want you to imagine these pathways you see, connecting them to the cell. Many students may just look at this image of a pathway and think of it like texts in a textbook, but you need to consciously think about, what does this mean if I enter into a cell? Where am I? So glycolysis, as we said, the process of breaking down glucose all the way to pyruvate, was happening in the cell cytoplasm. Once we are arriving at pyruvate, if we have oxygen available, then we enter into the mitochondria. And this is then, here is where you have to use the illustration of the Krebs cycle. And imagine that all of that is happening in the mitochondria. However, before we move on to the Krebs cycle, we just talked about the option of aerobic conditions, meaning oxygen is available, but what if oxygen is unavailable? So what if we have anaerobic conditions present? In that case, pyruvate will be converted into lactate, and the enzyme is lactate dehydrogenase. Now, let's look at the citric acid cycle, also known as the Krebs cycle or the TCA cycle. It is again happening in the mitochondrial matrix. What is the process? If you remember, looking at this cycle, very often you will see an illustration like this. You need to now know that there was an arrow pointing into acetyl-CoA. That was glycolysis. No, that was pyruvate. So essentially, we are saying that there was an aerobic condition here for pyruvate to go into the Krebs cycle. So the process is pyruvate is converted into acetyl-CoA, which enters the cycle. Then it produces NADH and FADH2. These are electron carriers and 2 ATP. They will release carbon dioxide as a byproduct. So the importance of this step is that it supplies high energy electrons for the next stage. So the Krebs cycle is like a power generator breaking down fuel to produce energy-rich molecules. Now, these high-energy electrons move then to the final stage, which is oxidative phosphorylation. So, oxidative phosphorylation, step 3. Now, this is happening in the inner mitochondrial membrane. So, we had glycolysis, right? We took the example of glucose. Going through glycolysis in anaerobic conditions in the cytoplasma. If arriving at pyruvate and we have oxygen available, it is converted into acetyl-CoA, entering the Krebs cycle with the purpose of producing high energy molecules. Now entering into step 3. So we have moved now from the cytoplasma first into the mitochondrial matrix. After that, we move into the inner mitochondrial membrane. Now, what's happening here? So the NADH and FADH2, the electron carriers produced in the TCA cycle, in the Krebs cycle, they donate electrons to the electron transport chain. And these electrons pass through protein complexes, creating a proton gradient. These protons then flow back through ATP synthase, generating ATP. So we have now arrived at the generation of ATP. What is the end result of this process? We will produce the majority of ATP here, which is approximately 34 molecules per 1 glucose. So oxidative phosphorylation is like a hydroelectric dam using the flow of protons to generate a large amount of energy, ATP. And this process, the electron transport chain process, requires oxygen. Oxygen acts as the final electron acceptor. This is also another point worth mentioning, is that this is also the difference between anaerobic and aerobic bacteria, where anaerobic does not have oxygen as a final electron acceptor, while aerobic does. Now, let us ask the question, what happens if oxygen is unavailable? So let's look at anaerobic metabolism. So anaerobic metabolism, the definition is ATP production. So we still produce ATP, but without oxygen. Now the process, as we mentioned, we had first glycolysis, arriving at pyruvate. If we have oxygen, pyruvate enters into the mitochondrial matrix, further into step 3, where we have oxidative phosphorylation, and the end result was 34 ATP. That's a huge amount for one glucose. But if we do not have oxygen, pyruvate is converted into lactate, and the end result will be 2 ATP per glucose. So you can see the difference, whereby one produces 2 ATP, the other one produces 34. So anaerobic metabolism is like running on backup power. It's not as efficient. It's actually way less efficient, but it keeps things going. Now let's briefly mention the regulation of metabolism. So enzymes that are involved in all of these different steps, like pyruvate dehydrogenase or lactate dehydrogenase, etc., 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 these enzymes are controlled by feedback inhibition. So for example, ATP inhibits glycolysis. The point of glycolysis was what? Break down glucose, get it to pyruvate, get it into the TCA cycle, produce more ATP, right? But what if we have a lot of ATP produced? We need to have some kind of system in place where our body receives feedback to stop glycolysis. So the overproduction of ATP will inhibit glycolysis, and this is a feedback inhibition system. And this is what works for enzymes. Another way our metabolism is regulated is by hormonal control. For example, insulin. It promotes glucose uptake and storage. Another one is glucagon. It stimulates glucose release from storage. So metabolism adjusts based on energy demand. Are you fasting? Are you feeding? Etc. Now, glucagon, is worth mentioning, is a hormone released by your pancreatic alpha cells. And indeed, it is to stimulate glucose release from storage. And insulin is also produced or released by your pancreas, but not by your alpha cells, rather by your beta cells. And they promote glucose uptake and storage. So in summary then, ATP is the primary energy carrier generated through glycolysis, the Krebs cycle, and oxidative phosphorylation. 2. Oxygen is essential for efficient energy production, but anaerobic pathways do provide backup. And metabolic pathways are tightly regulated to balance energy supply and demand. Now, from now on, I want you to always visualize these different steps. When we say glycolysis, step 1, the Krebs cycle, step 2, oxidative phosphorylation, step 3. I do not want you, like many medical students actually do, which is they miss the idea to visualize this as it is happening physiologically in our body. So when you hear glycolysis, immediately try to connect it to a cell. Imagine a cell. Imagine the cell cytoplasma. And this breakdown is happening of glucose into pyruvate is happening in the cytoplasma. Now you need to think a step further. You have an organelle here called the mitochondria that you study in your initial courses. Once it enters there into the mitochondrial matrix, this is where we now know then oxygen was present. So we're in the Krebs cycle now. And here these high energy molecules are produced, the carriers needed for step 3. And that was happening in the inner mitochondrial membrane. And when we go to number 2 here, oxygen is essential for efficient energy production. Yes, because this step, right, these steps provided us with approximately 34 ATP, and anaerobic pathways through the production of lactate gave us 2. And that's the end of lecture 8. Continue now to chapter 9.